Assalamu alaikum wa rahmatullahi wa barakatuh. Bismillahir Rahmanir Rahim. Welcome to today's biology class. Uh, Welcome to the second class of chapter 4, Bioenergetics. In the previous first class, we have discussed and understood the concept of bioenergetics and its field of discussion. We said that bioenergetics is the field of biology, especially is a sub-branch of biochemistry, where the chemical energy conversion is discussed in detail, and its field of discussion in your book is only limited to photosynthesis and cellular respiration. We have understood the role of ATP and other high energy compound. ATP is the main high energy compound, and NADP, NADH, FAD, they are the secondary high energy compounds which provide necessary energy to perform different chemical reactions inside the cell. We have understood the phosphorylation and dephosphorylation cycle that undergoes in the chemical ATP always. We have understood why is ATP called the biological coin, as ATP is a universal chemical substance that can be present in every cell and can provide energy to perform any kind of energy intensive chemical reaction. We have also defined the photosynthesis process. We have also discussed about the 4 requirements of photosynthesis and also understood its chemical reaction. And the site of photosynthesis in plants is the mesophyll tissue of the leaves, and the oxidation and reduction process takes place simultaneously in the photosynthesis reaction. We say carbon dioxide is reduced and water molecule is oxidized during photosynthesis. In today's class, second class, we will discuss about the 2 phases of reactions that takes place during photosynthesis. From the knowledge of previous classes, we came to know about the photosynthesis reaction as a single step reaction having only a single chemical equation. But actually the photosynthesis reaction is performed after a series of reactions, more than 50 successive steps. So the photosynthesis reaction is completely performed after completing a consecutive steps of more than 50 chemical reactions, and there's more than 50 chemical reactions are categorized and grouped into 2 phases. The first phase is called the light dependent phase, and the second phase is called the light independent phase. We will discuss about them in details later on. And during the light dependent phase, 2 important chemical reactions takes place. They are called photolysis and photophosphorylation. We will understand what is called photolysis and photophosphorylation, and why is called ATP and NADPH as assimilatory energy. Because ATP and NADPH, they are produced during the light dependent phase. They helps in the light independent phase to capture carbon dioxide, to assimilate carbon dioxide to produce glucose. And the process of generating glucose by carbon reduction in C3 cycle, we'll understand about the C3 cycle in detail, and we'll know how carbon dioxide is captured and this carbon is reduced to produce glucose. We'll understand them in detail. At first, we understand what are the 2 phases of photosynthesis. The 2 phases of photosynthesis are the first phase is light dependent phase that takes place in thylakoid, and the second phase is called the light independent phase that takes place in stroma. That means a set of chemical reactions takes place in the thylakoid, and to perform this set of chemical reactions, presence of light is necessary, and using the light energy from the sun, the chemical reactions in this phase can take place. That is why these sets of chemical reactions are collectively included in the light dependent phase, while the later chemical reactions after the light dependent phase, The later chemical reactions, these steps doesn't require the presence of light energy, and to perform these chemical reaction steps, uh, the light energy from the sun is not used. That is why the later chemical reactions are included in a phase called light independent phase, which takes place in the liquid matrix of the chloroplast called stroma. That means light dependent phase takes place in the thylakoid membrane we see here, and the light independent phase reactions take place in the stroma, in the liquid matrix part of the chloroplast. Now we will see a diagrammatic procedure how light dependent reactions take place in the chloroplast, and then it is followed by the light independent reactions in the stroma. We will show these consecutive steps with diagram. At first, assume that this is a double membraned chloroplast. Inside the chloroplast there is thylakoid. Each of the disc is called thylakoid, and the stack is called granum, and the liquid part is called stroma. And in the thylakoid there are numerous number of molecules present on the surface, and these molecules are collectively called photosystem. As we see in the previous figure, here we see on the membrane of thylakoid there are numerous number of molecules. These different molecules collectively forms the photosystem. And what are the components of photosystem? Important components. Here we see the chlorophyll, and these two molecules are electron transporter, electron carrier. This one is NADP reductase enzyme. This one is ATP synthase enzyme. These are the important components of photosystem. And how the light dependent phase starts in the thylakoid. We know from the sun light energy comes in form of photon, and then it falls on the chlorophyll, and using the light energy the chlorophyll first breaks down water molecule that is present inside the thylakoid membrane. So the chlorophyll uses the light energy to break down this water molecule. Here we see H2O. The water molecule is broken up. That means water molecule has been splitted up using the light energy in the chlorophyll. This process is called photolysis. What is the literal meaning of photolysis? Photo means light, and lysis means breakdown. We understand using the light energy, if we break down water molecule to produce one oxygen atom, two protons and two electrons, this process is called photolysis. And then these two electrons and two protons will go to the NADP reductase. The proton will directly travel to the NADP reductase, while the two electrons will travel to NADP reductase using the electron transporter carrier molecules. And also in the stroma there are two high energy compounds present called NADP plus and ADP. They are not at the highest energy state, they are at some lower stage, but they can acquire energy, they can capture energy to form NADPH and adenosine triphosphate. So the NADP plus present at the stroma will go to the NADP reductase. So at the site of NADP reductase enzyme, three molecules come: NADP plus, protons and electrons. And the NADP reductase enzyme facilitates a chemical reaction to produce NADPH plus H plus. Here NADPH is called the reduced nicotinamide adenine dinucleotide phosphate, which is a important high energy compound, and the rest of the proton is not attached with the NADPH, but that freely floats in the stroma. Here we see the NADP plus absorbs one proton and two electron to produce reduced NADP, and the single proton that freely floats in the stroma. So now we see a high energy compound has been produced. And also there are numerous number of ADP molecules, adenosine diphosphate molecules, present at the stroma. This ADP molecule will attach with a phosphate group. This phosphate group is not seen in the figure, but you should assume that a single phosphate molecule is attached with adenosine diphosphate, and that comes to the ATP synthase enzyme to perform a chemical reaction to produce adenosine triphosphate. That means the ATP synthase enzyme facilitates the reaction to attach a inorganic phosphate group with adenosine diphosphate to produce adenosine triphosphate. And this process of production of adenosine triphosphate is called photophosphorylation. From the name we can understand that photo means light and phosphorylation means adding a phosphate group to adenosine diphosphate. That means we understand what is called photophosphorylation. Photophosphorylation is the process of adding a phosphate group with adenosine diphosphate to produce adenosine triphosphate using the light energy from the sun. So these two, photolysis, photophosphorylation and production of NADPH, these are the three important chemical reactions that takes place during the light dependent phase. And after the completion of light dependent phase, this produced ATP and NADPH then helps to perform the chemical reaction of light independent phase, that is called C3 cycle. The light independent phase is performed by the C3 cycle in most of the plants, but some of the plants don't follow the C3 cycle during the light independent phase. They follows either C4 cycle or CAM cycle. We will discuss about them later. But what are we saying? That the energy of ATP and NADPH is used in the C3 cycle to produce glucose. So these cyclical chemical steps starts from the RUBP that absorbs carbon dioxide molecule. So the C3 cycle starts when RUBP, ribulose 1,5-bisphosphate chemical, absorbs carbon dioxide molecule to produce a keto compound. Then this keto compound transforms into 3-PGA, 3-phosphoglyceric acid. What is the 3-phosphoglyceric acid is the first stable compound of the C3 cycle. Then this 3-PGA converts into G3P, glyceraldehyde 3-phosphate, and during this conversion the energy of ATP and NADPH is used to facilitate this conversion to G3P. So the ATP and NADPH serves the necessary energy to produce G3P, and by releasing the necessary chemical energy they converts again into adenosine diphosphate and NADP plus. And again this G3P recycles to produce RUBP, but during this recycling process one molecule glucose is produced. And we see that the RUBP, the first precursor chemical compound, is again generated, and this chemical will again absorb some more amount of carbon dioxide and then again produce keto acid, then 3-PGA, then G3P, then again RUBP, and again ATP and NADPH will be used up to produce glucose molecule. And this cycle continues until there is a steady supply of carbon dioxide and ATP and NADPH assimilatory energy. And now, why is ATP and NADPH called assimilatory energy? They are called assimilatory energy because they helps to assimilate carbon dioxide to produce glucose. What is called assimilation? Assimilation means to make something, to convert something as a part of its own. So to assimilate the carbon dioxide, the energy of ATP and NADPH is used. That is why the energy of ATP and NADPH is called the assimilatory energy. So now we'll understand the same topic we have discussed in the previous slide. We discussed that light dependent phase takes place in the thylakoid, and two important reactions during the light dependent phase is photolysis, there is a breakdown of water, and photophosphorylation, production of ATP, and NADPH generation, that is the third important chemical reaction during the light dependent phase. And we have already discussed about why is ATP and NADPH called the assimilatory energy. And then we have also talked about light independent phase that takes place in stroma, and in the stroma actually carbon reduction takes place, and this carbon reduction takes place by C3 cycle in most of the plants, but some plants follow the C4 cycle, and again some plants follow the CAM cycle for carbon reduction to produce glucose. Now we will see a video, the same content in form of an animation.
Photosynthesis. Within eukaryotic photosynthetic organisms, also known as photoautotrophs,
The light photons are falling on the chloroplast cells.
of photosynthesis occur within plant cells in specialized structures known as chloroplasts.
And
Photosynthesis consists of two sets
light
of
photons
reactions,
are falling on the
light-dependent
chloroplasts.
reactions and the Calvin cycle. Within the
Inside the chloroplast we see there are thylakoids, a multiple number of thylakoids. Multiple number of thylakoids produce granum, and the liquid part is called stroma.
chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma.
And we know that the light dependent reactions take place in the thylakoid, and the light independent reactions take place in the liquid matrix stroma.
The reactions that synthesize glucose, the Calvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic,
These are the components of photosystem present on the thylakoid membrane. Here we see there are two photosystems present, photosystem 2 and photosystem 1. These green colored parts are filled with chlorophyll, and these spherical shaped molecules are electron transporter, and the last of it, this part is NADPH reductase, and this machine like component is the ATP synthase enzyme.
organisms, thylakoids contain pairs of photosystems called photosystem 1 and photosystem 2 that work in.
Now we will start how the chlorophyll breaks down water to produce oxygen, electrons and protons.
The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons.
Water molecules come to the chlorophyll, and using the light energy, the chlorophyll breaks down the water molecule into 2 protons, 2 electrons and 1 oxygen atom. And in this process, if 2 oxygen atoms are formed, 2 oxygen atoms merge together to produce single oxygen molecule.
Electrons and oxygen gas.
And this process is called photolysis, we know already. And by this photolysis process, oxygen is generated, which is actually the source of production of oxygen during the photosynthesis process.
While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways.
Now the electrons will travel through the electron transporting molecules or carriers, and ultimately will reach to the NADPH reductase.
As electrons pass through the electron electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase.
We have also discussed that the ATP synthase enzyme produces adenosine triphosphate by combining a phosphate group with adenosine diphosphate. So here we see ATP synthase enzyme is working to produce adenosine triphosphate,
which phosphorylates ADP to form ATP. The low energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the...
And we have said that the electrons come to the NADP reductase enzyme, and also protons come at that site, and NADP plus that also comes. So what happens? NADP reductase enzyme combine these three things to produce
NADP plus to NADPH.
to produce NADPH. So now you see what is going on the thylakoid membrane. We see photolysis at the chlorophyll, NADPH production on the NADPH reductase enzyme, and ATP production on the ATP synthase enzyme. By this process, numerous number of ATP and NADPH is produced, and they freely float on the stroma to supply the necessary energy for C3 cycle to produce glucose.
A steady supply of photons.
And we have already said that to perform the chemical reactions on the thylakoid membrane, we need light energy, but to perform the later chemical reactions on the stroma, we don't need to use the energy of sunlight. The energy will come from the captured from the energy stored inside adenosine triphosphate and NADPH. That is why the energy stored in ATP and NADPH is called the assimilatory energy.
NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions.
This Calvin cycle reaction is performed in most of the plants during the light independent phase, that is also called C3 cycle.
The Calvin cycle consists of a series of reactions that...
Now we will discuss in detail about the Calvin cycle or C3 cycle in the later slide.
Photosynthesis.
So what is called C3 cycle? C3 cycle is also called Calvin-Bentham cycle after the names of the discoverer scientists. As for their important discovery, they were awarded Nobel Prize in 1961. Why is this cycle called C3 cycle? Because these chemical reactions takes place successively in form of a cycle, and the first stable product produced during this C3 cycle is 3-carbon phosphoglyceric acid. 3-carbon phosphoglyceric acid molecules, this is the first stable product that contains 3 carbon. That is why this cycle is called C3 or carbon 3 cycle. Most of the plants follow the C3 cycle. We have said that already. Now how the C3 cycle starts? The C3 cycle starts from the precursor molecule called RUBP. RUBP in full form, that is called ribulose 1,5-bisphosphate molecules. There are millions of RUBP molecules present in the stroma in a chloroplast. But for simplicity, we take 6 RUBP molecules to start the cycle. So 6 RUBP molecules, precursor molecules, attach with 6 carbon dioxide molecules. In other words, they capture 6 carbon dioxide molecules to produce 6 unstable keto acid molecules. And during this process, an enzyme called Rubisco helps for this capture process. Uh, that is why it is called the carbon fixation step during the C3 cycle. And then these unstable keto acid molecules then breaks up immediately to produce stable compounds, 3-phosphoglyceric acid. It produces 12 3-PGA molecules, 3-phosphoglyceric acid molecules. So from 6 keto acid we get 12 3-PGA molecules. And then this 3-PGA phosphoglyceric acid molecules will convert into G3P glyceraldehyde 3-phosphate molecules. But this conversion requires high amount of energy. This energy comes from expanding adenosine triphosphate and NADPH. We are saying that the energy of adenosine triphosphate and reduced NADP is used to produce G3P glyceraldehyde 3-phosphate molecules. That is the energy intensive step of the C3 cycle. Then among these 12 G3P molecules, 10 G3P molecules are put apart for recycling process, and 2 G3P molecules are taken apart for production of glucose. So 2 G3P molecules are taken apart to combine into a 1 glucose molecule, and the rest of the 3 G3P molecules are recycled to produce again the precursor molecule RUBP. And during this process also the energy of ATP is consumed. So by this process, what we have noticed? We noticed that the chemical reaction started from the RUBP molecule. It captured the carbon dioxide molecule, produced keto acid. This keto acid is unstable, that produces the stable product 3-PGA, 3-phosphoglyceric acid. This 3-phosphoglyceric acid is converted into glyceraldehyde 3-phosphate molecule, and during this process the energy of ATP and NADPH is used, and this step is called the reduction step because according to the electronic definition of reduction, we know if any chemical accepts electron, that is called reduction. So here we see the NADPH has released electron, and this electron is accepted by the G3P molecule. That is why this phenomenon of receiving electron by the G3P molecule is called the reduction process. And among the 12 G3P molecules, 2 G3P molecules are set apart to produce 1 molecule of glucose, and 10 G3P molecules are kept aside to regenerate into again the 6 RUBP molecules. That was the precursor molecule. That is the regeneration step. And this cycle continues indefinitely as long as there is a steady supply of carbon dioxide and steady supply of assimilatory energy molecules. So at the end of today's class, what we have learned, let's revise. We have understood the 2 phases of reactions in photosynthesis: light dependent phase and light independent phase. During the light dependent phase, a set of chemical reactions takes place, and among this set of chemical reactions, photolysis and photophosphorylation are important. Photolysis is the breakdown of water molecule, and photophosphorylation is the formation of ATP by the ATP synthesis enzyme using the light energy from the sun. And we also know why is ATP and NADPH called the assimilatory energy, because they provides the necessary energy to assimilate the carbon dioxide to produce glucose during the light independent phase. We also know the process of generating glucose by the carbon reduction in C3 cycle. We know the C3 cycle in detail. At the end of today's lesson, you have a homework. Explain the 2 phases of photosynthesis in C3 plants with diagram figure 4.04. This figure is provided in your book. So সবাই ভালো থাকো ইনশাআল্লাহ। সুবহানাকা আল্লাহুম্মা ওয়া বিহামদিকা আশহাদু আল লা ইলাহা ইল্লা আনতা আস্তাগফিরুকা ওয়া আতুবু ইলাইক। আসসালামু আলাইকুম ওয়া রাহমাতুল্লাহি ওয়া বারাকাতুহু।